prior to the 1950s, was considered a non-pathogenic E. coli. It didn't cause disease. But then starting in was it 1970s, physicians, microbiologists started isolating these E. coli from these horrible cases of bloody diarrhea. And in some cases, the patients were continuing to develop kidney failure and a hemolytic anemia. And who's responsible? This strain of E. coli. And I'm like, what the heck's going on? This guy isn't supposed to be making toxin. So what microbiologists now believe is that a close cousin of E. coli called Shigella dysenteria, this is the bacterium that causes bacillary dysentery. This is the original bacterium that made Shiga toxin and carried the Shiga toxin gene. So it's believed that at some time the Shigella dysenteria was infected by a temperate phage, okay, so plus some temperate phage that integrated itself into the Shigella's chromosomal DNA, and at some time there was induction, so that there was DNA damage to the Shigella. The phage cut itself out of the Shigella chromosome, but accidentally cut into the chromosome itself, carrying the Shiga toxin gene with it. So it went into the lytic cycle. We had replication of the phage DNA and the Shiga toxin gene. That combination of phage, Shiga toxin, DNA was packaged into new phage that were released. And one of those recombinant phage, so we call them transducing phage, infected which bacterium? That non-pathogenic E. coli 015787. Remember, years ago, this E. coli, it didn't make toxin. It was benign. It was a non-pathogen. So that transducing phage through specialized transduction then transferred the Shiga toxin gene to this E. coli. Okay? And the phage DNA and the Shiga toxin DNA got inserted into the E. coli chromosome. Now, the reason they know this, when they do the DNA sequencing of the toxin-producing E. coli 015787, they find the DNA sequence for the Shiga toxin gene right next to the DNA sequence for prophage. So we see that the specialized transduction, it is a powerful force in which new emerging bacterial pathogens have evolved. New toxin-producing strains of E. coli, for example. And this means, you guys, that in your lifetimes, you too will be witnessing the evolution of new bacterial pathogens, new toxin-producing strains of maybe E. coli. So this is a powerful force in the evolution of new pathogens. And again, we see that viruses are the delivery trucks of the donor bacterial DNA to new recipients. When we start talking about medical microbiology, we'll start talking about these powerful protein toxins that many bacterial pathogens produce, and you'll be amazed at how many of them were delivered by specialized transduction. They're carried by prophage. It's just mind-blowing, mind-blowing. Okay, so we'll keep going, guys. So just so y'all know, this whole topic, it's just delicious subject for compare and contrast type questions on the lecture exam, the next lecture exam coming up. So classic questions are compare and contrast lytic bacteriophage to temperate bacteriophage. Compare and contrast the lytic replication cycle to the lysogenic cycle. Compare and contrast generalized transduction to specialized transduction. And on D2L in the supplemental notes, I tried to make a couple of tables, just giving examples of how you can organize the information. Like, for example, which steps in the lytic cycle are similar to the steps in the lysogenic cycle? Which steps are different? So just remember that this often ends up being the topic of multiple choice questions, or it could be part of the short answer section where I have you fill in a table comparing and contrasting. Yeah.
So is the E. coli 015787 technically infected with a virus? With the
Right, it is a lysogenize bacterium. And I I don't know, this is information I don't know, but I'd love to know. Last May, so almost a year ago, in May of 2011, there was this new Shiga toxin producing strain of E. coli that maybe you guys remember. They had an outbreak in Germany, it spread to Northern Europe, and this one was called E. coli 0104H4. And it surprised everybody because it's a powerful Shiga toxin producer. And what I don't know is how this strain acquired its Shiga toxin gene. What I don't know is, was it specialized these are just, I don't know the answers. Was it specialized transduction starting with this lysogenized 0157? You got induction, you got cutting out the Shiga toxin gene, and then that recombinant transducing phage infected this other strain of E. coli? Was it this similar story all over again, where we had a Shigella dysenteria that was infected? And those are, and I'm sure the really smart microbiologists are doing all the DNA sequencing, so probably the answers are out there. I just haven't found them yet. Yeah. So, I don't know if this guy can act as a reservoir of Shiga toxin gene to be spread via specialized transduction of Enterobacteriaceae or not. If it was Shigella to here, and maybe for the end of the semester we'll find out the answer. Yeah, it's very disturbing. Very disturbing. Yeah. When these lysogenized bacteria, where the profiles are actually carrying these toxin genes with them, it's very disturbing. Yeah. So, okay. So we use bacteriophage replication not only to help us understand the basics of viral replication, but also to help us understand how viruses will continue to play an important role in the evolution of new bacterial pathogens, right? Okay. Now what we want to do is we want to turn to viruses that can infect animals, including us humans. So humans, we want to remember we are animals too. And what we'll see is we'll keep the basics of bacteriophage replication in the back of our minds, and we'll see how do human viruses, how are they similar in the way that they replicate to bacteriophage? How are they different? So we're going to launch into just a little introduction to animal-human virus replication, and just look at some of the challenges, why it took us so long to understand, how human viruses replicate compared to these bacterial viruses. So first of all, because again, viruses are obligated intracellular parasites, it was a lot harder to study animal or human viruses in the lab because the ethical issues of purposely inoculating animals, and certainly we had advanced so we weren't inoculating humans with viruses to test them out. So the ethical, moral issues there. And also using live animals as experimental models is very expensive. There was a big leap forward when it was discovered that many viruses can grow in fertilized embryonated eggs. So that was a big help. But there were problems with that too. Not all viruses will grow in fertilized embryonated eggs, and it takes a lot of expertise to be able to inoculate the eggs properly. There's problems with the eggs becoming contaminated with bacterial or fungal contaminants, and then your experiment is shot. There was a huge breakthrough in the 1950s with a technique called cell culture or tissue culture, and it was discovered that many more viruses could be grown in cell or tissue culture. And I'll just give you like a simplified cartoon of this cell or tissue culture technique. So in the old days, you would have special sterile little glass or plastic tissue culture plates, and you would have growth media for your cells. And in some of the simplest experiments, what you do is you'd harvest some tissue from an animal, or maybe it was, like a human, grind it up with some of the growth media, and then you place that cell mixture in your tissue culture dish. And if you were lucky, what would happen is that the cells from that homogenized tissue they would attach to the bottom of the plate, and then they would start replicating, forming a monolayer. So if you were successful, you'd have this nice monolayer, a single layer of cells. And again, these could be cells, say, from an animal, it could be cells from a human. And then what you could do if we said, we'll just use little asterisks here to say this is our virus, you could inoculate your tissue culture cells with a virus, and if you were lucky, the viruses would be able to attach and infect the cells. And so after incubation, after you let the viruses replicate, they would be released from the infected cell, and then you can harvest the growth mediator, which would now hopefully be chock full of viruses. You could use those viruses, for example, to do vaccine studies. One thing folks would do is they would have numerous repetitions of these virus-infected tissue culture plates and add different drugs to see if one of the drugs could block replication in a search for antiviral drugs. So this was a big step. This tissue culture permitted the growth of many more viruses in the lab, but it too has drawbacks. It turns out that some cells, for example, human liver cells, will not produce the receptors for some of the hepatitis, the liver-infected viruses. So, for example, you can't grow hepatitis B virus in tissue culture. You can't grow hepatitis C virus in tissue culture because the liver cells won't express the correct receptors, right? So, again, still have some problems, but it was a huge breakthrough. So this is just some figure showing figure 13.17 on page 397 and figure 13.18 showing some of the virus cultivation techniques. This shows an embryonated fertilized egg, and it's showing all the different ways the egg can be inoculated. And again, we see here the expertise that's involved. Some viruses will only grow in the amnion. Other viruses will only grow on the chorioalin membrane. So again, a lot of expertise. Not everybody was good at infecting these eggs. And again, not all viruses would grow in embryonated eggs. This is a modern example of tissue or cell culture where these flexible bags are used. You have a monolayer of cells in here. This would be the growth media. You have these ports through which you can inject your viruses. You can inject drugs of interest, and then ports from which you can withdraw the growth media to harvest any of the virus that have been produced. So again, these techniques help tremendously in the study of replication of human viruses. Okay, so what we're going to do, you guys, is we're going to look at some of the steps of human and animal virus replication. And what we're first going to do is look at how animal virus infect our cells. And I'll use animal human virus interchangeably. So we saw with the bacteriophage how the bacteriophage would inject their nucleic acid into the host cell. So we want to see, was that how animal viruses do it? And it turns out the answer is, well, some of the animal viruses use that model. So for example, polio and dengue viruses, just to get you oriented here, this represents the surface of the human cell right here. So this is the cytoplasm, this is cytoplasmic membrane. These little pink things are the surface receptors to which the viruses will attach. Okay, so one way that viruses such as polio and dengue virus will infect our cells. Here we have the protein capsid with the nucleic acid. The adhesins on the capsid bind to the surface receptors, and then the capsid forms a pore through our cytoplasmic membrane, a hole through which the nucleic acid will be injected into our cells. So that looks pretty similar to bacteriophage, right? But there are other strategies which other viruses use to infect our cells. And this next one, I really like. I don't like. Well, it's of interest. Okay, this next way of infecting cells is used by measles, mumps, and HIV. We're going to be looking at HIV replication next week, so we'll come back to this. And this is a technique that envelope viruses can use, and it's called fusion. Now remember, the envelope of the virus is stolen host cell membrane. It's made of the basic, the same composition as our cytoplasmic membranes. And remember, the consistency is like olive oil. So here with our, this might be HIV. We see in the envelope the HIV is inserted its proteins, some of which are going to be adhesins. Those adhesins bind to special surface receptors on our cells. Now, what then happens is the viral envelope melts or fuses with our cytoplasmic membrane. Just fuses right into it, melts right into it, and that permits release of the protein coat, the capsid, into the cytoplasm. The protein coat, the capsids, are made of these little subunits called capsomeres. They are disassembled, releasing the viral nucleic acid into our cytoplasm again while we're infected. So that's called fusion. This last technique is also very cool. This is a technique that's used by adenoviruses, herpes viruses, and influenza viruses. This is kind of the Trojan horse technique. The viruses trick our cells into endocytosing, bringing the virus into the interior of our cells in a membrane-bound vacuole endosome. So here we've got, this could be, I'll make this up. Let's say this is influenza virus. The envelope influenza virus, it binds to surface receptors on our cells. And again, that tricks our cells into endocytosing the virus. So here we see that invagination of the cytoplasmic membrane, formation of the endosome here. And here we have the virus that's now being carried in the endosome. What happens, for example, in influenza virus is once this endosome has been formed, the virus has been brought into the cytoplasm, it then triggers fusion of the virus envelope with the endosome membrane, and that's what we have right here, with release of the capsid. The capsid subunits are disassembled, releases the viral nucleic acid. So to me, this is like the Trojan horse. You're kind of tricking the cell. It's like, oh, I got a great present for you. Yeah, it's like, uh-oh. So Trojan horse approach here. Okay. So different viruses have different techniques of infecting our cells. Okay, so we'll see that some of the basic steps then are very similar to bacteriophages. First step is always attachment, okay, or absorption. Then, and this, I should have put that up for you guys, I do apologize. There should be step one up here, should be attachment or absorption. We have entry, in which the viral genetic information is introduced into our cells. And then we have synthesis of the viral genetic information and proteins. Now, usually, but not always, RNA viruses are replicated in the cytoplasm, and usually but not always, DNA viruses are often replicated, the genetic information is replicated in the nucleus of our cells. But remember, you guys, viruses love to violate our rules. They keep you very humble, viruses do. So there's always exceptions to these rules of where the viral nucleic acid will be replicated. Following replication of the viral genetic information and synthesis of the viral proteins, we're going to have assembly, just like we assemble a car. And then there'll be release of the viruses. And there's different ways that viruses can be released. Now, one way is similar to the living cycle we saw with bacteriophage. It might be that the host cell itself will be lysed, it will be broken open, the cytoplasmic membrane will be broken, releasing the newly replicated viruses. That's one technique that some naked viruses use. They just cause lysis and release of the newly replicated viruses. Some viruses will escape by the opposite of endocytosis. They'll trigger the cell to release them by exocytosis. And then this last technique, to me again, is really a very kind of cool technique. This is called budding. And budding is a common way that envelope viruses steal some of our cell membrane. So this process of budding, we see in this cartoon, and again, we could pretend maybe this is HIV. HIV uses budding to escape from our cells. So again, to get you oriented, this would be the cytoplasmic membrane of our cell. Here's the virus that's been partially assembled. We have the capsid that's been formed around viral genetic information. Now, this virus is going to, if this is HIV, the HIV is going to steal part of our cytoplasmic membrane. Okay, before it does, it inserts viral proteins into our cytoplasmic membrane. Some of these will act as adhesins. Okay? And what we see is that the viral capsid migrates over to the cytoplasmic membrane, and it almost looks like it's pushing against it. And this causes an evagination, a protrusion, almost like a bubble that's being formed of the cytoplasmic membrane. And this continues until finally the virus escapes from our cell, taking with it a coat of the cytoplasmic membrane that then becomes the envelope. Right? So this process is called budding, and again, it's a common way that envelope viruses acquire their envelope by stealing our cytoplasmic membrane and budding. Pretty cool stuff. All right, now going back to that, gosh, replicating that viral DNA, this gets a little bit messy, you guys. So we will try really hard to keep it as simple as we can. Okay. So if we had a DNA virus, meaning the viral genetic information is DNA, which enzyme would the virus use to make copies of its DNA? That's just a general name.
DNA polymerase.
DNA polymerase, right, okay? And, you know, that's usually not a problem because our cells have DNA polymerase, right? That's one reason that many DNA viruses replicate their genetic information in our nuclei. They just use our DNA polymerase. Some viruses encode their own DNA polymerase, but a lot of viruses just use our own DNA polymerase in our nucleus. Yeah, not a problem, right? But think, you guys, what if you are an RNA virus? Okay, so let's think about this. Let's say that you are a single-stranded RNA virus. So your genetic information consists of a single strand of RNA. Which enzyme are you going to use in a human cell to make copies of your RNA? Okay, and that is such a good thought. That is such a good thought. Oh, and I'm so glad you said that. Okay, because we're going to, and this is what gets kind of messy, you guys. We're going to go into some naming strategies here. So think of cellular RNA polymerase, rRNA polymerase, and let's think about this, you guys. Let's make a little table. And in our cellular RNA polymerase, what is the template? Meaning, what is the guide? What is our RNA polymerase use as a guide to make RNA? What is it binding to?
Single-stranded DNA.
Okay, in transcription, you guys remember we said DNA acts as a template to make RNA, right?
Uh-huh.
This is in the cell, right? So our cellular RNA polymerase uses what as a template to make complementary RNA?
DNA.
DNA, dynamite, you guys. Okay, so for the template, we would say that cellular RNA polymerase uses DNA. And when we start naming these enzymes, we can get fancy. We can say cellular RNA polymerases are what we call DNA dependent. This just means it's just telling us what the template is. DNA dependent, and then we ask what is making. So what does the enzyme make? It makes RNA, right? So in the second part of the name, we put what it makes, RNA, and then we just add polymerase because this is a strategy for naming enzymes, right? Okay, so it turns out that our cellular RNA polymerase are what we call DNA-dependent RNA polymerases. They're RNA polymerases that have to have single-stranded DNA as a template, right? Okay, so let's back up, you guys. So could an RNA virus use our cellular RNA polymerase to make copies of its RNA? Could the cellular RNA polymerases, which require a DNA template to make RNA, do you think our cellular RNA polymerases could use this RNA as a template?
No.
It can't. It can't. So it turns out these viruses require a special viral RNA polymerase. And how do you think we're going to name these special RNA polymerases? What's the template? There you go. You nailed it, you guys. So the viruses have to supply their own RNA dependent. And what are they going to make? What's the viral RNA polymerase going to make? RNA, right? So these viral RNA polymerases are RNA dependent. They have an RNA template, and they make RNA. So the take-home point here, you guys, is do our cells make RNA-dependent RNA polymerases?
No.
No, we don't have any need for it, right? So the RNA viruses, they must encode information for their own RNA-dependent RNA polymerases because our cells don't have it for them, right? So that's kind of a challenge. Okay, so these RNA-dependent RNA polymerases, sometimes they're called viral transcriptases. Sometimes you'll see them described as viral transcriptases. Okay. All right. So when an RNA virus infects a cell, it either is going to bring with it preformed RNA-dependent RNA polymerase, or what will happen is the RNA that is introduced will be transcribed and translated into an RNA-dependent RNA polymerase. Okay, and it gets really complicated. Don't worry about it, okay? But what I want you to tell me here is, for these RNA viruses, they inject or introduce into our cells these single-strand RNA templates. What will be the enzyme that is going to make the copy? So now we have double-stranded RNA. What's the name of this enzyme that carries out this copying process?
Viral
that is
transcriptase.
that viral
Transcriptase.
Right, yeah, and I'll just put the full name just so we can practice it. That's a viral RNA-dependent RNA polymerase. Okay, so that's a unique enzyme that most RNA viruses have to supply the information for, because the host cells, the animal cells, aren't going to have it, right? Okay, now, just to make our lives crazier, guys, but this will be the end of craziness, hopefully. It turns out, you see how we have, down here we have double-stranded RNA?
Mhm.
Well, it turns out when you have double-stranded RNA, you remember that this strand is a complement to this other strand, right? They get labeled as the plus and minus strands. So you want to understand what the heck do they mean by plus and minus strand. So I always want to have something really simple I can remember when I'm taking an exam and I'm exhausted, right? So what I would do whenever I was taking a virus exam, right when I got my exam, right at the top, I would say plus or positive single-strand RNA equals mRNA. What that means is that the plus strand acts as messenger RNA. Host ribosomes bind to it and translate it into viral proteins. Okay. Does that make sense, you guys? Just put a little box on this, and on that next lecture exam, just as soon as you get the exam, just write it at the top of the page. So we could say, for example, this is the plus strand. So what do you think this strand is? It's
Negative.
the complement. It's considered the minus or the negative strand. Okay. So the minus or negative single strand RNA, this is going to make you crazy, is the complement of mRNA. Okay. Now, just so we can play with this the tiniest bit, you guys. Okay. So if we're talking about RNA viruses, single-strand RNA viruses, so they have single-strand RNAs that are genetic information. They get split into two groups. Those that have positive or plus single-strand RNA, and those that have negative single-strand RNA, or minus single-strand RNA. So when these plus or positive single-strand RNA viruses, when they inject their cells with their RNA, you can say, oh, that's not a problem because that's plus single-strand RNA. It's going to act as messenger RNA. Our ribosomes bind to it, they translate into viral proteins, and voila, the virus is off to the races, right? Not a problem. But what's going to happen if you have a negative or minus single-strand RNA virus, and it injects that negative single-strand RNA into our cells? How will it make messenger RNA? which enzyme must it carry with it that is going to get introduced into our cells at the same time the virus is introducing its genetic information. How can we go from a negative or minus single-strand RNA to get a plus single-strand RNA or messenger RNA? There you go. You got it, you guys. Okay, so the virus has also to deliver when it infects us the viral RNA-dependent RNA polymerase. And with that, it'll take the negative or minus single-strand RNA as a template and make the plus strand. And remember that plus strand then is going to act as what? That's going to act as the messenger RNA, and our ribosomes will then bind to it and translate it. Now I know that just seems like gymnastics, you guys, and it's cruel and unusual punishment to do that to you right before the lab practical, but it is really important for us to understand how those RNA viruses, they require these unique enzymes. And sometimes what will be done if a patient presents with some unknown infectious disease and people are like, what the heck's going on? Sometimes they'll assay for these unique viral enzymes as a clue to at least let folks know, is this a viral disease? Is it a bacterial disease? Is it a fungal disease? So certainly if you were to find evidence of RNA-dependent RNA polymerases in an ill patient, you're like, well, we know they've got an RNA virus on board. Whether it's the cause of the disease or not, we don't know. But we know they've got an RNA virus on board. Okay. All right, you guys. So I think that was the messiest part, you know, and I do understand a lot of people think the nucleic acid stuff is kind of boring. So hopefully now it'll get a little bit juicier. All right, so again, talking about different types of viruses, you all remember we said that in the bacterial viruses, we had the lytic phage that would always kill their host cell, and then we had the temperate phage that may or may not kill their host cell. And we're going to see human viruses, there's different types, kind of the same differences in behavior. So some viruses will always kill their host cell, their animal or human host cell, and they are called lytic animal viruses or cytocidal. Cytal means to kill, cyto refers to cell. And so these guys, very often they are naked viruses, okay, because they don't need to steal cell membrane. All they want to do is get out of the host cell so they can cause lysis to release, you know, massive numbers of viruses. And so this is an example of a cytocidal or lytic virus, adenovirus. Here we see it's a naked virus. It has these very cool spikes, little protein spikes that act as adhesins. This is electron micrograph. You can just barely make out some spikes. And this is a result of an adenovirus infection where you have massive inflammation and breakage of blood vessels that might be a result of lytic infection with the adenovirus. So those are pretty dramatic. Now, just as we saw, well, this would be unfair to say that these budding viruses are like lysogenic viruses. That's not really true. Okay, so we just saw an example of a lytic or cytocidal virus that causes lysis of the host cell. The host cell dies. And if we were to do an experiment with a cytocidal virus like adenovirus, and we were to graph the number of infectious virions in the growth media on the y-axis against time, okay? And let's say in our little experiment we're doing this in tissue culture. So we inoculate our tissue culture cells with a certain number of viruses, and then we start taking little aliquots of the growth medium and we start assaying for the number of infectious viruses that are present. Well, what's wild is initially they seem to disappear, but they haven't truly disappeared, right? They've invaded the host cells. And we've got this period here where the viruses are inside the host cells, they're undergoing synthesis and assembly. And with those lytic or cytocidal viruses, what will happen is it will get this lysis of the host cell and release of thousands and thousands of newly replicated viruses. So we get this burst of viral release during lysis. Now, the second strategy that many viruses use, including HIV and influenza virus, is, remember HIV and influenza, they are envelope-viruses. They're going to escape by budding through the cytoplasmic membrane. So if we repeat that experiment with, say, HIV or influenza virus, again, we see the disappearance of the viruses when they infect the cells. This is a lag period where we have biosynthesis and assembly. But remember, with those budding viruses, they are escaping by stealing little tiny bits of the cytoplasmic membrane. They don't initially cause our cells to break open. They're not cytocidal initially. Okay? So what happens is you slowly get release of those viruses as they're budding away from our infected cells. And that means our cells remain infected potentially for long periods of time. So our infected cells with these budding viruses can remain alive, producing viruses for long periods of time. We don't get the big jump, the big spike in replicated viruses, okay, but slow release. Now, often the host cells that are infected by these budding viruses, eventually they become exhausted because all the resources are being redirected to make viruses. But initially they won't die. They live and continue to release virus for a long period of time. So these budding viruses, sometimes people refer to them as causing persistent infections, where you have low level of virus release over a long period of time. Okay, so the infection persists. You can remain infectious for long periods of time because the viruses are slowly being released from our cells. Now, on a happy note, we want to say, well, why is studying the virus replication? Why is that important? Well, it's crucial in helping us to understand how we can stop or control the spread of viruses. So one important concept I want you guys to understand is how important it is for viruses to attach to the surface of our cells before they can infect them. So this is just our little cartoon, say, of a human cell, cytoplasmic membrane, and these little U-shaped guys are supposed to represent the surface receptors of our cells to which the virus will attach. Okay. So let's take a look at this virus right here, and here are the little viral protein spikes, and these little balls at the end of the spikes would be the adhesins that are going to bind the complementary surface receptors on our cells to initiate infection. Well, a powerful way that animals have evolved to protect themselves against viruses is animals can produce proteins called antibodies or immunoglobulins. And here we see the little cartoon. This is a generic cartoon for an antibody or immunoglobulin. And you guys might remember way back in cartoon chemistry, we said kind of our generic antibody we use as an example is called IgG, immunoglobulin G. It's the predominant antibody in humans. And we describe how it's made of 4 polypeptide chains that are arranged in roughly a Y-shaped structure. And so you guys might remember we said there's like a stem, which would be kind of like our legs and our torso. And then it has these 2 arms, just like we have 2 arms. And at the end of the arms are little very specific clefts called antigen binding sites. And it's these antigen binding sites that permit antibodies or immunoglobulins to bind to invading pathogens or bind to their toxins that they produce. Now, the beauty of these antibodies is if they bind to the adhesins, they block attachment. If antibodies bind to the surface of the virus, if antibodies bind to the adhesins can't bind to our cell surface receptor. So these antibodies block attachment. We have a fancy name for antibodies that block attachment. They're called neutralizing antibodies. So these neutralizing antibodies are powerful ways that our body protects us from viral infection. So neutralizing antibodies. So neutralizing antibodies, they block attachment of pathogens or toxins. Neutralizing antibodies are often would give us lifelong immunity against some pathogens. And in the last part of the course, we'll talk about specific immune responses, right? Neutralizing antibodies are one of the things we're trying to trigger in our children when we vaccinate them. We're trying to trigger our children to produce neutralizing antibodies against specific pathogens or toxins. So if they ever encounter them in the real world, these neutralizing antibodies will bind to the surface of the invading pathogen or the toxin, block attachment, and then that pathogen, that toxin, can cause harm. So almost finished here, you guys, with this introduction. So another topic we want to address is, why is it that viruses are so difficult to treat? Okay, why is it they're so difficult to treat? So we'll just take a look at a couple of reasons. Well, one is, because viruses replicate inside our cells, that provides a protected niche for them. Most of our antiviral drugs are water soluble, they're hydrophilic. So to reach the viruses when they're being replicated, the drugs must pass across the cytoplasmic membrane and possibly into the nucleus, so there's more cell membranes to cross. Each of those cell membranes is a diffusion barrier. So just replicating inside our cells, it's much harder to get the correct levels of drugs inside our cells than it is to get correct levels of drugs in the extracellular environment. So the viruses are replicating in a protected environment inside our cells. The second reason, and perhaps more important, is you want to remember the viruses are using our biosynthetic machinery to copy themselves. So to stop viral replication, it often means we have to stop biosynthesis, normal biosynthesis within our own cells. And the problem with this is many of the antiviral drugs we use, it would be great if we could only shut down biosynthesis in the virus-infected cells, but our drugs aren't that specific, right? And if we give our patients systemic drugs, those drugs will shut down normal processes in infected and non-infected cells. So often it's a race. Can we win the race? Can we stop the viral replication before we cause significant damage to our patients' own cells? And often the really unpleasant side effects of taking antiviral drugs is because we're causing harm to our own good cells. So that's a huge issue. We just want to remember that one group of antiviral drugs are the nucleoside or nucleotide or base analogs. These are look-alike drugs, look similar to normal nucleotides, nucleosides, bases, but they will either, when they're incorporated by DNA polymerases or RNA polymerases, they'll either trigger mutations or they'll cause premature termination of nucleic acid synthesis. So they stop RNA synthesis or they stop DNA synthesis. And again, the goal is to stop viral replication, but these analogs will be used by our uninfected cells as well. So we can see inhibition of normal DNA replication. We can see inhibition of transcription, and again, that is going to cause damage to our good cells as well. This last tidbit, you guys, is latency. This is a phenomenon seen in some viral families, for example, the herpes virus family. One of the, we had a guest speaker from UC Davis. He was a medical microbiologist emeritus, very distinguished gentleman, and he asked the class, he said, "Okay, class, why are herpes viruses like diamonds?" And I was totally perplexed. I felt so bad I didn't know why herpes viruses were like diamonds. And so he says, very straight face, he goes, "Because herpes are forever."
Herpes are forever.
You guys can start the Thursday with latency. They will describe what we mean by that. And